Hello guys, welcome back to our YouTube channel, The Ignited Medicos. Today we shall learn about the functions of glutamic acid, aspartic acid and asparagine. Glutamic acid, it is a non-essential and glycogenic amino acid. It donates one proton to form glutamate, and glutamic acid undergoes ATP dependent reaction in the presence of glutamine synthase to form glutamine. Both glutamate and glutamine play a predominant role in the amino acid metabolism and are directly involved in the transfer of amino group for urea synthesis. Now let us study about the specialized products formed from glutamate. First one, glutathione. It is a tripeptide that contains glutamate. Second one is the N-acetyl glutamate. It is an allosteric regulator of carbamyl phosphate synthase 1, which is the first enzyme in urea synthase. Third one, glutamate is present in clotting factors, namely 2, 7, 9 and 10 as gamma carboxyglutamate and is involved in coagulation. Fourth one is the GABA, that is gamma aminobutyric acid. The glutamate, it is decarboxylated in the presence of glutamate decarboxylase to form GABA. This is a sensitive and irreversible reaction. It is catalyzed by PLP, that is pyridoxal phosphate dependent glutamate decarboxylase. Now let us study about the metabolism of GABA. GABA undergoes transamination reaction in presence of gamma glutamate transaminase to form succinate semialdehyde. Succinate semialdehyde undergoes oxidation in presence of succinate semialdehyde dehydrogenase, requires NAD plus as a coenzyme to form succinate. Succinate then enters the TCA cycle. This serves as an bypass for glutamate to enter the TCA. Now let us study about the functions of GABA. It is one of the major inhibitory neurotransmitters in the brain, and GABA regulates the activity of neurons by discouraging the transmission signals. The mechanism of inhibition. It is believed that GABA opens chloride channels and increases the permeability of postsynaptic membrane. Thus, GABA functions as an inhibitory neurotransmitter. Vitamin B6 deficiency and GABA. GABA synthesis requires pyridoxal phosphate, a coenzyme of vitamin B6. In vitamin B6 deficiency, the production of GABA is reduced. This results in neuronal hyperexcitability causing convulsions. Now we shall study about glutamine. Glutamine is the most versatile amino acid and it is the temporary storage form of ammonia. Glutamine is highly diffusible and hence it is easily transported. It is the donor of nitrogen atom for purine and pyrimidine synthesis and it takes part in many conjugation reactions. Now we shall study about aspartate and asparagine. Both these amino acids are non-essential and glycogenic in nature. Aspartate is formed from oxaloacetate by transamination reaction in presence of AST, that is aspartate transaminase. Aspartate has certain important functions, namely it donates one amino group for the synthesis of urea, and aspartate forms a link between urea cycle and TCA cycle. Aspartate is utilized for the synthesis of purines and pyrimidines, and aspartate takes part in malate aspartate shuttle for the transfer of reducing equivalents, namely NADH, from cytosol to mitochondria. Now let us study about asparagine. It is synthesized by aspartate by a synthetase in an irreversible ATP dependent reaction. Asparaginase hydrolyzes asparagine to liberate ammonia. L-asparaginase is an anti-cancer drug against leukemias and lymphomas. Thanks for watching.